are regulated. In your human body cells, you can have proteins that can bind to certain gene regions to increase the rate of transcription for the transcription enzyme RNA polymerase. Or you can have proteins decrease transcription to the point that it may not be transcribed at all. This is a form of gene regulation. Your eye cells don't use the portion of DNA that codes for making hydrochloric acid like your stomach cells do, because there is regulation in your cells to determine which portions of the DNA is used. But we want to shift gears now to talk about a very interesting way of regulating genes that can be sometimes a little challenging to visualize. This particular method is really found in prokaryotes, a few eukaryote exceptions. It's called an operon. An operon is a fancy way of regulating genes, and it's usually made up of a few genes that can involve enzymes. Remember that enzymes are proteins with the ability to break down or build up the substances that they act on. Now let's talk about some key players in an operon so that we can see some gene regulation here. First, RNA polymerase. It's a builder enzyme actually, because RNA polymerase is an enzyme. In biology, a lot of enzymes end in that ASE. RNA polymerase is needed in order to start transcription. Remember that transcription and translation are steps in protein synthesis. Protein synthesis means to make proteins, enzymes in this case. The thing about RNA polymerase, though, it gets a little confusing for RNA polymerase without somewhere to bind. If you watched our DNA replication video, you learned about DNA polymerase and how it had to have a primer to know where to start. Well, RNA polymerase needs a promoter. A promoter is a sequence of DNA where the RNA polymerase can bind. So you would think, okay, you got your RNA polymerase, you attach it to a promoter, and boom, you make your mRNA, which eventually will be used to make a protein. But there's this other sequence of DNA called an operator. The operator is a part of the DNA where something called a repressor can bind. The repressor, if bound to the operator, blocks RNA polymerase. And poor RNA polymerase cannot move forward and no mRNA can be made, therefore no proteins. So let's take a look at our setup here. This is an example called a lac operon. Notice there's a promoter region of the DNA, the operator region of the DNA, and there are 3 genes here that code for enzymes that help in the process of breaking down lactose. Lactose is a sugar. If lactose sugar is around, bacteria want these enzymes to be made so that they can use them to break down the lactose sugar. Then they can metabolize it. Fed bacteria are happy bacteria. Here's the repressor. There's actually a gene here that codes for producing the repressor. See this gene that we call I? It has its own promoter. Well, this gene codes for the production of the repressor. So why do we need this repressor? Well, it's wasteful to make things that you don't need. If there's no lactose, it wouldn't make sense to start making enzymes that work together to break down lactose. It would be a waste. The enzymes would just sit there. So if lactose is not present, then the repressor binds to the operator, and this blocks RNA polymerase. So mRNA cannot be made, and therefore the proteins, the enzymes in this case, cannot be made. But if lactose is around in the environment, something pretty cool happens. The lactose, remember that's the sugar, binds to the repressor. This changes the repressor's conformation.
Really fast.
Try as it might.
So it just said it changes the conformation of the enzyme. That just means it changes its shape. And like we've talked about how shape is super important to function. Now that that's bound together, that repressor can't even fit on the operator anymore. Like it won't fit on it, so it can't bind. And that's why. You can hit play.
The repressor cannot bind to the operator. RNA polymerase finds its promoter, binds, and transcribes to make the mRNA from the genes on the operon. That mRNA will be used to make enzymes to break down the lactose sugar. And bacteria like to eat, so that makes them pretty happy. We have to say that we think it's pretty impressive to think about all the gene regulation that occurs in cells. And if you find it fascinating, know that there are careers that focus on gene regulation and understanding how genes can be turned on and turned off. Because by doing that, we can gain a better understanding of a variety of diseases that have gene influences in the human body. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Yeah, it's pretty cool, right? Probably not. You guys?
It's amazing.
So, yeah, that's Nopron. There's some other cool, interesting things with gene regulation. It's like really, really complex, you guys. So like, remember yesterday we were watching the video with the annoying guy, whatever his name is, and he mentioned like how do we get, if we're an embryo, like where do we get all these important things we need? Remember we get them from mom's egg cell? And so depending on the contents of that egg cell, what's in it, that can have a huge influence on our genetics. It's like a huge environmental influence. We call them maternal factors. And so just depending on whatever egg ends up being fertilized, it just, the contents of the egg cell can create a completely different environment for gene expression to be regulated in different ways. And then probably the most important thing that I want you guys to take away from thinking about gene expression, because it's something that affects most of us in our lives, either ourselves or someone we know, is cancer. And so when cancer happens, we've talked about how it's just cells going wild, growing out of control, continuously dividing, and not going through those checkpoints that like make cells not divide anymore. And so that happens when there's mutations in genes, like something that controls for like a repressor or a silencer, for instance. So if a certain gene is supposed to be turned off and then some mutation happens where now that gene isn't turned off and can't be turned off anymore, you're going to get a cancerous cell and probably get a tumor, which is bad. So that's kind of how cancer works, and it has a lot to do with gene regulation. And so now we're going to talk about mutations, which are exciting and fun. So there's a few different types of mutations we're going to talk about. The first one, it's called a point mutation, and literally all it is, so this is a DNA strand right here. It's just a substitution of a base. So they took this C and accidentally, or wait, this C, I'm sorry. They accidentally put an A. Who probably accidentally put it there? This happens during DNA replication. No. So there's no RNA involved in DNA replication. But what's that really important enzyme that synthesizes the DNA? Starts with the word DNA. Polymerase. Yeah, DNA polymerase. Remember, so DNA polymerase like makes the mistake, but also fixes the mistake. Remember when it proofreads, goes back over things? So this is like it's probably the most common type of mutation. And most of the time it's fixed, right? Remember, DNA polymerase can proofread, it can go back, it can fix it. However, it does miss some mistakes. And as we get older and older, those mistakes start to build up. And so mutations like this are like a huge part of aging and dying, basically. Eventually, like when you're 80 years old, most of the cells in your body have more mutations than they did when you're, what are you guys, 15, 16?
Yeah.
Right? Yeah. And so when a point mutation happens and there's a base, yeah, Riley.
Well, I had a discussion with my friends that kind of related to this. So your cells start replicating when you mate. So once your cell has replicated long enough, doesn't it like completely change from the original cell?
So yeah, if it's a stem cell, so if it's like a very beginning cell that can differentiate into any type of cell type, that's true. But for instance, like a skin cell, which rapidly divide all the time, right? Because we're losing skin cells, like dead ones, all the time. After a while in the cell cycle, many cells, especially if they like discover they have all these built-up mutations and things, they'll go into programmed cell death, is what it's called. It's called apoptosis. It's like a separate part of the cell cycle, kind of on the side, where they'll be like, oh, like, you're not functioning right anymore, like, you have too much wrong with you, like, go kill yourself, basically. That sounds bad, but that's what happens with the cells.
That's the worst job.
Yeah, yeah, Tammy.
Isn't it something like your skin completely replaces itself like 900 times in your life or something like that?
I mean, I don't know exactly, but I think it's like every 7 years you literally have all brand new cells in your skin. Like they're not the same ones that were there before, which is cool. But yeah. So anyway, though, you guys know that if we change the sequence of DNA, it's going to change that mRNA sequence, which is going to change what? What other sequence? What's the ending sequence it's going to change? Yeah, the amino acid sequence in your protein's going to be wrong. And then if the amino acid sequence isn't right, is the protein going to fold correctly? No, it's not. Also, every once in a while, you can get a mutation that adds in a base pair that creates a stop codon. So it could be a perfectly fine mRNA, but then the ribosome hits that stop codon, and then it doesn't make the rest of the amino acid sequence, the polypeptide. So you're gonna have a shortened protein, and is it gonna fold correctly? No, it's not. It's not gonna be functional. So that's one type of mutation. And a lot of times, so when we look at a codon, you guys remember how, so one amino acid can have multiple codons that code for it, right? So yesterday we saw like leucine has 6 different codons that code for leucine. Usually it's that third base that can change, and so that's called base pair wobble. Base pair wobble. And so if there's a mutation and it changes that last base in a codon, a lot of the times it's not going to matter. It's not even going to change the amino acid. It's going to just be the same thing. And when a mutation doesn't actually change how the gene's expressed, we call it a silent mutation. Like it just exists there, but it's not actually doing anything bad or good. It's just there. So you can go to the next page.
There, but it's no longer active.
Kinda. Yeah. I think the ruler disappeared, so you're good. So then the next kind of mutation is an insertion or a deletion, where instead of like a base pair being swapped with the wrong one, there's actually just a base is inserted where it's not supposed to go. And so remember we talked about the reading frame of an mRNA and how like it matters what order these are in because we have to get the right codons for the right amino acid. And so if we insert a base pair, it's going to shift that reading frame over. And as you can see here, it happened. It shifted it over. We don't get the right amino acids because we're not reading the code the right way, like it's supposed to be read. Does that make sense to you guys? Yeah? Okay, cool. All right, and then, so mutations like that are usually happening during DNA replication. But during meiosis, when we make our sperm and our egg cells, chromosomal mutations can happen where chunks of chromosomes can accidentally like get onto a different chromosome. This is displaying gene duplication where, so these would be homologous chromosomes. They're like, let's just say this is chromosome 1. Like they're both chromosome 1, but they're your 2 different copies. Yeah. So something happened where an entire gene got duplicated on one chromosome and then the other chromosome's missing that gene. And that can be not good if we have not the right amount of genes that were supposed to on one chromosome accidentally ending up on the other one, right? And so go to the next one. And so another thing called gene translocation can happen where a gene from one chromosome goes onto a completely different chromosome that it's not supposed to be there, which obviously is not going to be a good thing. And so why it's not going to be a good thing is because remember there's all that like regulatory DNA that controls gene expression. So if we just like take a chunk of chromosome and separate it from all the stuff that's like above it on the chromosome, we might be separating it from its promoter or its like operator region, and then you can't control gene expression for it, and it's going to lead to a lot of problems. Yeah. Any questions about this? No? All right. So, talked about this. Mutations usually don't lead to problems, but they can if they build up enough. or if they end up in a part of our DNA that's important enough where we can't have mutations. Yeah, you can go to the next one. Okay, we already talked about this, and so, yeah, we already talked about that. Right, it's really important to remember that the mutations in our body cells, so like, if we get cancer, we can't spread that, right? We don't pass that off to our offspring. Only mutations that happen during meiosis will pass on to our offspring, or if, like, so, if you get like a bunch of x-rays in your pelvic region, that can cause mutations in your eggs and sperm, and then that's when those mutations will get passed to offspring. But just normal, everyday mutations that happen from DNA replication, you're not going to pass those on. However, if you guys are interested, I mentioned this to the other class, in Tasmanian devils, they actually have a form of cancer that is contagious. They spread it to one another, it's on their face, and so like if they're nuzzling each other or if they're fighting and like biting each other, they'll spread this face cancer to one another, and it's like a very big problem for their population. Yeah.
So then how can you spread it? If you have it, then how can you spread it? Like, they spread it.
Well,
Like,
say
it's
if you have a
it's
kid.
it's not like any normal cancer, right? Cancer is not contagious, but this one is like crazy, and it
Yeah,
is.
they all have that cancer, so they
Well, not all of them have it.
Great.
So like they can spread it to devils that don't have it.
Grace, it'd be like if they had a kid, because you can't pass on cancer to a kid. You can pass on the chance, but you can't pass on
Right.
it itself. So the kids wouldn't have it at birth, but then say if the parent nuzzled them, then the kid would get it.
Yeah, it's just really cool, really complex.
What kind of animal is it?
But Tasmanian devil.
Oh, those things are real. Like that thing
Yeah,
from Looney
they're
Tunes?
interesting.
No.
But-
That's right.
Okay. And so, why are mutations important? Like, mutations are important, you guys. Like, life would not exist without mutations because mutations are the entire basis of natural selection. Literally selecting for advantageous mutations. Because every once in a while, maybe some organism gets this, like, a mutation that creates a whole new enzyme that does some cool new thing in their body that makes it so they outperform all the rest of their species. And then from that, it can lead to an entire new species being created. So, mutations are important. They drive evolution. We can go to the next one. Okay, and so we've already talked about what causes mutations. Number one thing, replication errors during DNA replication. Also, we've talked about mutagens like UV rays, radiation. There's also lots of chemicals that cause mutations. And so this gal, really cool scientist, Rachel Carson, she wrote this book called Silent Spring, which, remember we talked about that chemical DDT and how it biomagnified in the ecosystem, and it caused eagle and osprey shells to like deteriorate, and so their populations declined. She was the woman who was like, hey, this is doing this to the environment. We have to stop using it. So she's awesome. She also brought to the forefront a lot of like saying like, hey, like there's all these other chemicals that they're going to cause mutations and they're going to cause cancer. So there's also one interesting drug that used to be used like 60 years ago, widespread use. It was used as it's called an antiemetic, so you don't throw up. It was given to pregnant women so they wouldn't get morning sickness, so they wouldn't throw up all the time. And so this was like the most major drug crisis that's ever happened in the world. It caused over like 10,000 birth defects in babies of women who took this drug to prevent morning sickness. And when I say birth defects, I mean it ranged from like mild to severe deformities of like missing limbs. So, do you guys want to see?
Ooh, yeah.
So you can see like this baby messed up, right? He doesn't really have arms, he doesn't have legs.
I don't know. That baby's
He's missing an arm. Like their hands, they'd get polydactyly, multiple fingers.
That'd
be cool.
Feet on his hand.
And at least 10,000 reported cases of severe like malformations like this. So that's a huge deal. And so once they realize like, oh my gosh, it's this drug that's passing on, like the chemicals in this drug are crossing the placental barrier, getting into like the embryo, affecting the fetus. So they don't prescribe it anymore. And if you, well, they prescribe it for certain things. They actually prescribe it to treat this certain kind of cancer. And it's an interesting drug. They don't actually know how it works very well, which a lot of times, you guys, like our medicines, sometimes scientists like don't actually know the entire mechanism of how they even work.
So you might die.
Yeah. That's true.
Yeah,
That's why there's a
commercials
side effects
that are
and
like,
things like
oh,
that.
try this medicine, and then at the end it's like,
Right. Or
warning signs
sometimes there's
and
medicines
death.
that were like made for one reason, and then we find out they actually work better to treat something else, which is pretty cool.
Say it really fast.
And so yeah,
so you don't
that's thalidomide.
Boy.
Big deal.
Side effects.
Pretty sad.
depression.
But so it caused like huge, horrible mutations in the fetus, is what was happening. And scientists, there's a bunch of different theories, but they think it had to do with like blood vessel formation. That's basically it.
Oh yeah.
All right, have a good day, you guys.
I'm gonna go.
Attendance thing.
The what?
Your picture, like your school picture.
Oh God.
Yeah.
I don't even know who put that.
I know. It's like you like